Thank you, Brian. And thanks to the organizers for having me. And also thanks to John Wakely for setting up so nicely some of the topics in coalescent theory that will be relevant to my talk. And so I just want to emphasize up front that this is a work that's theoretical and mathematical, and often when I'm thinking about organisms such as caterpillars or the lodgepole tree here, I'm thinking about mathematical abstractions based on those biological objects. So just to let you know that a lot of the trees and structures we'll be seeing will be mathematical ones. Though I will have a couple of organisms in the talk, but all the pictures are not from my lab. Okay. So if I might suggest that phylogeography and coalescent theory resemble a little bit of an island migration model with perhaps increasing rate of migration between them, ideas and methods going between them. We saw a little bit earlier in Anna's talk using methods, approximate Bayesian computation, and thinking about the site frequency spectrum, ideas that originally came out of coalescent theory in a very phylogeographic context. And so one of the areas that has been of interest to me has been a set of questions that have organically developed both in the phylogenetic or phylogeographic world and in the coalescent theory world, structured around this question of, under assumptions of a population genetic model, what properties do we expect for shapes of gene genealogies? So what properties do we expect in relation to population genetic parameters when we sample gene genealogies for multiple species? So a couple of areas that have been of interest to me, the concordance of gene trees, discordance of gene trees in relation to species trees, relationship of coalescence times to divergence times of populations with or without migration, and Scott had a nice paper with Peter Beerley on this topic. And then the topic that I'm going to spend this talk on, monophyly, paraphyly, and polyphyly of genealogical lineages sampled from multiple species. And since there are a number of different definitions of monophyly around, just define what I mean by it for this talk. If we have a genealogy here, I'll say that the lineages of species A are monophyletic. Here, in this case, the lineages of species A comprise all the descendants of their most recent common ancestor, which is right here, and can therefore be said to be monophyletic. And, of course, in this picture, the lineages of species B are monophyletic as well. Okay, so if we took lineages sampled from two different species, there's 4 different ways we could classify the genealogies for lineages from 2 different species. Here we have reciprocal monophyly. Both lineages from A and B are separately monophyletic. Here, paraphyly of B. The lineages of A are monophyletic, but the lineages of B are not monophyletic. So they don't comprise all the descendants of their most recent common ancestor, because some of those descendants are in A. The reverse case, paraphyly of A, and then polyphyly, where neither species has monophyletic lineages. So from a coalescent point of view, it's very natural to ask, what are the probabilities that genealogies have each of these different shapes? So we have our set of possible shapes. If we want to study random shapes, and John very nicely described why random shapes are of interest and where the randomness comes from, we add a coalescent theoretic model and then pose the question, what's the probability distribution of tree shape under that coalescent theoretic model? And this question really goes quite far back. It's been of interest for a variety of different reasons. These probability calculations on tree shape have appeared in quite a number of contexts, and I'm just listing some of them here. Theoretical predictions of patterns in genealogies, so just trying to understand what we might expect to see if we sample genealogies for multiple species. Estimation methods that rely on tree shape. hypothesis testing about population divergences and gene tree discordances, what's causing gene tree shapes to have the shapes they have. Species delimitation, so using monophyly to identify species or population groups. A whole mathematical area on combinatorics of evolutionary trees uses a lot of these tree shape problems. And then an area that's of increasing interest now, comparing genomic regions from different parts of the genome or different types of markers. We heard a little bit earlier this morning about mitochondrial DNA versus autosomal loci, as well as identifying genomic regions with unusually recent or ancient divergences that might indicate something interesting about speciation or natural selection. So the very earliest papers in coalescent theory were already thinking about probability distributions of genealogical shape. And Tajima in 1983, in one of the first coalescent theory papers, had this diagram with two lineages sampled from a species A and two from a species B. Could be just regarded as populations, not necessarily species. And he computed the probabilities under a very simple coalescent model of these four different shapes, which don't quite line up exactly with monophyly, paraphyly, and polyphyly, but are pretty close. And then looked at how those probabilities changed over time. Similarly, in a very early phylogeographic paper, Nigel with John Avise looked at the same kinds of calculations, here using a forward time simulation. So that explains the bit of wobbliness in the picture. And also, again, looking at four different shapes. So these kinds of questions have persisted over a fairly long time period. And I want to just review a couple of results from some of my studies leading up to the work that will be in the Colloquium paper. Okay, so if we think about the Tajima and Nigel and Avise work, the Tajima study used sample sizes only with sample size 2, and the Nigel and Avise work was a simulation. So I wanted to look at an exact probability computation of monophyly, paraphyly, and polyphyly in two species in order to have a more general way of looking at gene tree shape. So the goal here is, if we just choose one out of the four shapes, reciprocal monophyly, the goal is to compute the probability that a set of lineages sampled from species A, and this boldface represents the sampling, and these gray ones are the ones that are not sampled, and a sample from species B are reciprocally monophyletic. And the approach for computing that probability is to sum over all possibilities for what configurations of random lineages are present at this species divergence. So we look at using the coalescent lineages going backward in time and coalescing, sum over all those possibilities, and then given the possibility, or given the configuration at this divergence time, look at the probability distribution of what happens in the ancestral population. So we sum over all possible ancestral configurations, a product of the probability of a lineage configuration and the conditional probability of reciprocal monophyly, given the lineage configuration. Okay, so here are just the answers for the formulas for the four different gene tree shapes using this kind of approach. And you can see that all four equations have the same kind of structure. There's a sum, a double sum, the first sum representing something happening on the left side, the second sum representing something happening on the right side, a product of two terms, one for the left side, one for the right side, and then a third term which represents something happening above the root. And so this set of equations reproduces the theoretical formulas that underlie some of the observations from those early papers. Okay, so a couple of results. So here's the theoretical probability distribution of gene tree shape for two species, here with a sample of size 30. On the x-axis, we have time running in coalescent time units, so units of n generations, where each of the two legs of the tree has a population, a haploid population of size n, and then the probabilities of the four different shapes. So the two different paraphyletic ones are symmetric, so they're sitting right on top of each other. In this case, unlike what John had described, where there's a very strong sample size dependence for the pedigree problem, there's not that much of a sample size dependence in these tree shapes, although larger sample sizes do slightly delay the onset of reciprocal monophyly. The time since divergence is much more important. By about 1.67 n generations, reciprocal monophyly is the most prevalent of the different types, and by 4.37 n generations, 95% of the genome is monophyletic, 99% by about 6n generations. So there's some predictions about how long it takes to get to reciprocal monophyly. And these kinds of calculations have been used in a number of studies. I've just picked one out here from Bill Berkey, where he was looking at species delimitation in bdelloid rotifers, so that's his picture. And what you can see in this sample from a lot of different locations is that some of these branches are very long. So he looked at lengths of these branches in these different sampled individuals to try to identify when he should call them different species. And part of that calculation was based on levels of within group divergence to between group divergence of lineages that relate to these monophyly probability calculations. And so after seeing a number of these, I started to worry that chance branching of lineages within populations in small samples could be a possible explanation for at least some of the cases of reciprocal monophyly. And I'm not picking on this study or saying anything really about it in particular, but it struck me as a slightly unrecognized possible concern in deciding when reciprocal monophyly was observed. Okay, so to explain that in a little bit more detail, suppose we have some lineages that we've a priori, that we're a priori calling different groups. So say we have group A, 4 lineages, group B, 4 lineages, and we just decided a priori that those lineages are going to be different groups. And if we saw that they had a reciprocal monophyletic genealogy, most of the time what we have in mind is that something like this might have happened, that there might have been population divergence, and that group A and B are monophyletic because they're on different sides of this population divergence. But it's also possible that there could be an unstructured population, and if we just make a random genealogy from those 8 lineages, we might happen to get one that's reciprocally monophyletic. So could an observation could apply to any observation of reciprocal monophyly or monophyly of one group be the result of a small sample size and chance branching in an unstructured population. So we had all the calculations in order to develop a little statistical test. So here's the probability of reciprocal monophyly under random branching in unstructured population for different sample sizes. And you can see that those probabilities get small very quickly. So it does mean that those sample sizes don't need to be too big before this explanation of chance branching can be excluded. However, if the sample sizes are very uneven, so here's a case where there's only 2 lineages from group A, we need quite a lot of samples, quite a lot of lineages from group B in order to exclude an observation of reciprocal monophyly as having been due to chance branching. You can see that as those sample sizes get larger from group A, the sample size from group B doesn't need to be nearly as large in order to reject chance branching as being the explanation for reciprocal monophyly. Okay, so here's an example of this one. I took this one from Kubatko and Gibbs and Bloomquist, and this is a case in rattlesnakes. And so they had a number of different hypotheses, a number of different cases where they'd observed monophyly or reciprocal monophyly in a study in a lot of different rattlesnake groups in the eastern U.S. And they had, these are the sample sizes for different pairs of groups. And these are different hypotheses that they're testing about whether there's reciprocal monophyly or monophyly only of one group in relation to another. And they had a number of loci in the study, and so you can accumulate these probabilities across loci, and with a decent sample size and a decent number of loci, really only in the tens, you get very small probabilities quite quickly. So this from this kind of study, one of the possible concerns is that you can already see in the comparisons that they're putting together that they're lumping more than one group into one group in order to do a test that's pairwise. Okay, so there's more than two rattlesnake species or subspecies of interest, and they get lumped into two groups in order to fit them into the calculation, the two species calculation. So this two species calculation requires lineages of many categories to be grouped into two categories. And so really quite a number of people, and there's at least about three here, kind of been asking me about, can we do this kind of calculation where multiple species are considered separately, where monophyly is determined for really an arbitrary number of species simultaneously. And just looking back at our table, one of the columns in this table is number of species, and all of these studies had really only been looking at two species. There's a couple of calculations with three in the literature, but really all the main ones have two. So can we look at probabilities of monophyly, paraphyly, polyphyly in models that have more than two species in them? And it gets complicated very quickly. So this is a very simple genealogy with three species in it. And whether or not a species is monophyletic in a genealogy from a mathematical perspective doesn't depend on it can be determined just by looking at the lineages that descend from that most recent common ancestor. So here we have A is not monophyletic, B is monophyletic, and C is not monophyletic. But it gets very complicated when we try to look at the pairwise status of the different pairs of species. So just excluding the C lineages, what we can see is that A and B are reciprocally monophyletic, A and C are polyphyletic, and B and C have paraphyly of C with respect to B. So really just with a very simple diagram, we have a very complex description of that diagram. Okay, now with two distinguishable species that are labeled A and B, remember there are four ways to classify the monophyly status of the lineages, monophyly, paraphyly of A, paraphyly of B, and polyphyly. When we get to three species, there's 41. Okay, so there's the number of different ways of describing that genealogy in terms of all the different questions you could ask about monophyly. There's a lot more categories to consider. The natural approach would be to try to do something using a multi-species version of the coalescent. And what I mean by that is that we have coalescent models running separately in each of a set of species, and we put them together on the branches of a species tree. So each of these different branches represents a population that's on the lineage, on the branch of a species tree, and we glue them together with a set of rules for what happens at the species boundaries. Okay, so we have coalescent properties, processes happening within each branch, and when we reach a species split, going backward in time, lineages from all groups descended from the split are now allowed to coalesce. So how to compute the probability of monophyly on a species tree for arbitrary sets of species? And so this is where David Bryant came in. And it's really nice to get to describe this work first here in Southern California, because the first nugget of the idea for how to think about this came when I was actually still a postdoc at USC. And at that time, David was in Montreal at McGill, and of course he's from New Zealand. And so the way to get from Montreal to New Zealand is to have a long layover at LAX. And so he came to USC for about 6 or 8 hours on his layover, and we spent a nice day talking about phylogenetics and coalescent theory, and came up with an inkling that maybe the probability of monophyly could be computed by a pruning algorithm on an arbitrary species tree. So that's really about as far as we got. It's just posing this question. Of course, pruning algorithms are quite common in phylogenetic problems. So if we think about pruning algorithms in problems that involve coalescent and species trees, they have a structure where if you look at each place where two branches join going back in time to an ancestral branch, there's some output on the ancestral branch. And if we want to compute the probability distribution of the output of the ancestral branch, it has a structure where there's a sum over all possible left inputs to that ancestral branch, a sum over all possible right inputs to the ancestral branch, probability of the left inputs, probability of the right inputs, and then the probability of the outputs given inputs. So this last bit is what happens on this branch given what showed up at the bottom of the branch. So it's recursive because if we want to compute this term, the way to compute this term is to apply the same equation, but one step farther down the tree. So the left inputs will then be this branch will be the two branches that come in from the bottom down there. And since the time that we started thinking about this, at least just the very beginning, there have been several pruning algorithms developed for gene tree computations on species trees, including computations of the time to the most recent common ancestor, several computations about allele frequencies at biallelic loci under different circumstances, including one that I worked on with David, gene tree topology probabilities and ranked gene tree topology probabilities. So can we compute the probability of monophyly by a pruning algorithm? And so Rohan Mehta, who's here at the meeting, is a student in the lab, and he's the one who figured out how to make this work. So if you think about what the computation entails, we can imagine a species tree, and we have two classes of lineages, a set of lineages S and its complement. So that's the remaining set of lineages, which we'll call C. And that pair of lineage classes has 4 possible configurations: reciprocal monophyly, paraphyly of S, paraphyly of C, and polyphyly. So that's the way to get around this 41 possibilities, is to just think about it in terms of 4 cases. And so Rohan not only figured out how to put it on an arbitrary species tree, but added a different feature, which is that the lineages need not be restricted to single species. So, in the earlier computations, going back to Tajima, the 2 classes of lineages whose genealogies are of interest are coming in separate populations, but you can already see here in this diagram that there's S and C lineages can be in the same population. Now, they don't have to be. We could do the classic computation as a special case, but you can do this more general computation as well. Okay, so there's going to be a third type of lineage required for the computation, which we'll call a mixed lineage. And mixed lineage is formed by the coalescence of an S lineage and a C lineage, or the coalescence of really any pair of lineages of different types. So S lineages coalesce to form S lineages, C lineages coalesce to form C lineages, but any other pair of coalescences is going to produce a mixed lineage. So that's a lineage who's got descendants that are in both species. Okay, so that's the key insight, is to add this third classification of lineages, which are going to be lineages ancestral to both types. Okay, so remember, this is the shape of a pruning algorithm. So now that's the pruning algorithm, and here's what it looks like in this particular case. We have, say, if we're looking at reciprocal monophyly, we have, say, a branch X. We've got the two branches below branch X. We've got outputs of some node of branch X, and then inputs coming from the left and from the right. And if we're looking at the probability of reciprocal monophyly, what has to happen on branch X is all the coalescences have to be within species. So all the coalescences have to be only blue lineages coalescing with each other, or only red lineages coalescing with each other, until we get down to a case where there's just one blue lineage and one red lineage, and then they're allowed to coalesce. So there's events that are permissible, and then there are events that are not permissible. And in order to compute the probability of reciprocal monophyly, we have to sum over all the permissible events that could happen on that branch. So we don't have to get down to reciprocal monophyly on any branch until we hit the root branch of the species tree. So this is a perfectly acceptable set of events where there have been some coalescences on the right side, some coalescences on the left side, but we still have more than one lineage left on the left, and they haven't, the final coalescence producing reciprocal monophyly hasn't yet occurred. Okay, so this is just a schematic of the recursion equation that gives this pruning algorithm. And I won't go through it in too much detail other than to explain the terms so that you can see the structure. So this is a probability of a vector of outputs at branch X. So it's the vector of outputs up there, and that's three numbers. So it's the S lineages, the C lineages, and the M lineages. This is the sum over the inputs, so sum over the inputs coming from the left side and the inputs coming from the right side, and that's going to be vectors of length 3 as well. The probability of the left inputs, the probability of the right inputs, and then a function that describes the probability of the outputs given the inputs. And that's the complicated part. And so there's about 6 or 7 different cases for figuring out how to determine that function. Okay, so two more points before we'll see some results. Okay, so to use this recursion, we apply it starting from the root of the species tree. And the root of the species tree is different because the root of the species tree ends with just one lineage. So coalescent process, all the lineages eventually coalesce, and that last lineage is going to be a mixed lineage. So on the root branch of the species tree, we have to have this output, one mixed lineage. And then at leaves of the species tree, the probability is going to be set to 1 for the sampled lineage count and set to 0 otherwise. So when we get down to the leaves, then we don't need to keep applying that recursion. We just plug in the sample size that's actually been obtained. Okay, so this gives an exact computation of monophyly on a species tree. Okay, so just a couple of example calculations just to show what we can do with this. So this is a case with 3 species, and we might have a question, which is to compute the probability of monophyly of the blue lineages. Maybe we're interested in these 2 blue lineages, and then 2 that are sampled from another group. And here's this probability of monophyly as a function of the tree height, and then you could see there's this additional parameter r, which modulates where the third branch joins in. So if r is equal to 1, then what happens is this branch gets very short, and then as time increases, there's a long branch that has 4 blue lineages and 2 red lineages. So eventually, those 4 blue lineages and 2 red lineages are going to coalesce, and it's fairly unlikely to get monophyly. Okay, sorry, that was the, I've reversed the cases. So that was the case where, so if R gets is sent to 1, then these branches shrink, and then we have a branch here where the 4 blue lineages and 2 red lineages can coalesce. And so it approaches, as time goes to infinity, the probability that 4 blue lineages are monophyletic in an unstructured population, because eventually the 4 blue lineages and 2 red lineages are going to coalesce down to 1 by the end of that interval. The other case where R is set to 0 has this fairly long branch, and the other case where R is set to 1 has this long branch on it. So really the main point here is that the behavior is more complicated. Okay, so as we increase the number of species and the number of parameters of the species tree, we don't just see the simple increase in the frequency of reciprocal monophyly as time increases. That behavior of the probability of reciprocal monophyly or monophyly of a particular class of lineages can follow any of a number of different trajectories, and this is just one of about 4 different cases that Rohan observed. Okay, now here's an example that I think directly relates to this problem in the literature of having to test pairs of species separately. So suppose this were a species tree and we were interested in monophyly of these blue lineages. So the goal is to compute the probability of reciprocal monophyly of the blue and red lineages. Using a kind of the 2 species approach, we might be forced to lump those 6 blue lineages together and compute reciprocal monophyly using a 2 species tree. So that's what the past calculation would have entailed. And then it makes it interesting to compare the probability of reciprocal monophyly in these 2 cases, one that allows the multi-species gene tree to be used, and one that requires them to be forced into a 2-species tree. Okay, so here's the probability of reciprocal monophyly. Reciprocal monophyly this is the case with the actual species tree. And then this is the case where the lineages are lumped into 2 groups. So what you can see is that the lineages being lumped into 2 groups exaggerates the probability of monophyly. So there's, in the actual species tree, these lineages persist up the tree, and they're less likely to all have coalesced by the root of the species tree, whereas in this case, they're all possible to coalesce on that leg of the species tree. So that means that there's potentially a major difference in the probabilities that are obtained in these kinds of calculations when using the 2-species version in place of the multi-species version. And then these are just 2 other examples that involve multi-species versions with different ways of lumping here into 3 groups on the left and here into 2. So we wanted to do a couple of quick examples. So the one that we've done most extensively involves data from Mays and Teosinte that comes from this paper by Hufford et al. And Jeff Ross Ibarra. So of course Mays descended from Teosinte, and so there's a couple of ancestral Teosinte groups, Parviglumis mexicana, then the land races of Mays, and then improved inbred lines. And so this is a tree that we got from Jeff Ross Ibarra's lab as being a best estimate of the tree in a coalescent perspective from a quite large genomic data set that they put together with the Mays HapMap project. So that species tree is produced from genomic data without considering properties of monophyly. So we can ask, does that tree predict the properties of monophyly that are observed? So can we test if the species tree's predictions about monophyly match between theory and data? Okay, so kind of skip the details and just kind of give a broad outline of the data set. There are sample sizes of the different maize attacks of the improved land races, and the two teosyntes were quite different, so we trimmed it down to samples that were even across the different groups, considering different sets of lineages from among 75, and considering samples of size 2, 2, 2, and 2 from the 4 groups. And we broke the genome into short segments that were based on the decay of linkage disequilibrium in maize, and then did some more pruning to leave about 6,000 segments, each of which had a gene tree estimated. And then for each of those gene trees, we assessed the genealogical shape for each of those genomic regions. And then finally, just returning to different ways of taking those subsamples, we looked at the average genome-wide monophyly probability across the various different subsamples. And here's the result. So here are the 4 classes of lineages. Here are the frequencies of monophyly. And then these are violin plots representing distributions across the 100 different samples of lineages. So this is the genome-wide monophyly probability average is shown as a circle, and the distribution is across the different subsamples of lineages. And then those are our theoretical values shown in the triangles. And so we were really happy to see that they lined up pretty well, and at least in 2 out of the 4 groups, they're sitting pretty close to on top of each other. Okay, so what this is saying is that the theoretical model of the species tree, the monophyly that comes from our computation using the 4 species model, comes very close to predicting what's actually observed in a very tedious and complicated genomic analysis. So we have a second example in progress, and this is one of the species that Scott studies, the white-winged fairy wren. And here the method is similar, but the question has a very different objective, which is to determine if the monophyly of a particular set of samples within species that's observed at particular loci is unexpected given the species tree. So it's a question more about unusual loci. And we can approach it in the same way, which is to compute monophyly probabilities for a family of proposed species trees. Again, here it's going to be a 4-taxon computation. Okay, so to summarize, you can imagine these sort of 2 populations of theoretical approaches in the literature. We have the population of monophyly probability calculations, and then this population of pruning algorithms that have come together in an admixture to obtain a pruning algorithm for the probability of monophyly on an arbitrary species tree. The approach we have allows exact monophyly calculations with many species. It doesn't require all the lineages of interest to be in the same group, permits more complex monophyly scenarios to be investigated, and it does reasonably well at predicting the genomic data patterns in a 4-taxon example from maize and teosinte. Thanks very much.